So I don't have to tell you that hand movements are very important. All of you are doing hand movements right now. So, and we seem to do so without being aware of how complicated it is to control hand movements. And we do hand movements in a range. The repertoire of hand movements is really very, very large, as you can see here. Now, we can become very aware of how complicated it is to control our hands when we try to engineer robots. And if you think of it, robots right now are very far from achieving the great dexterity of the human hand, right? So my point is that if you understand the brain better, and if you understand how, maybe that's not up to date, and sorry, I'm a brain scientist, so I'm not really always up to date about all the best robots that are up there to simulate hand movements. But if you understand the brain better, and if you understand better how the brain controls hand movements, perhaps we could design better robots. And of course, we could design better brain-machine interfaces for patients who don't have the use of the limbs, so we can track which bits of the brain we can pick signals from to control robots and neuroprosthetics, right? So hand movements can be decomposed into two main components: reaching, grasping. Reaching relies on processing the distance, the direction of where the object is in the space, and then grasping, this is what we say in our lab, that it's about the size of the object, the shape of the object, orientation, and weight as well. Now, this is an old picture from the late Marco Davare, who studied grasping movements, and typically what people would do is they put two sensors on the tip of the thumb and index finger and the wrist as well, and what you do is measure the grasp aperture, and of course when you reach and grasp an object, the grasp aperture, maximum grasp aperture, is modulated by the size of the object. That's very basic. What we do now in our lab is we have a virtual reality environment to study grasping movements. So what we do is we have here a 3D screen, so you wear goggles so you can see things in 3D on the table. That's projected, the image on the screen is projected on the table via this mirror here. But what's underneath the mirror is our virtual world. It's basically two phantom robots, two force feedback robots, and they can push on your fingers based on how we program them. What we do is, this is the virtual scene that the object actually sees. So we want the object to see a cube, and via the robots, you can actually feel the cube and grasp it and lift it. And I've got a video here of what's happening in the lab. So that's me in the lab. I'm using TMS. I'm going to explain you what this is. So screen, mirror, the robots. You can see that the thumb and the index, the fingertips are represented by these two pink spheres. You can see them better the next trial. So these are your fingertips, and you grasp the cube and you lift it up and you put it back. In such a way, I can, in a very easy way, manufacture all sorts of objects. I can have small objects that are very heavy, so induce a conflict between what you see and what you feel, and that's great to understand multi-sensory integration. How do you integrate vision and haptics, vision and tactile cues? And we understood from this morning's talk that you can't guide a robot with a webcam, right? You need to have the feedback at your fingertips. So this is very important for controlling robots. Right. Now, we're going back here, and I'm a brain scientist. What I want to know is how does the brain process that information about reaching and about grasping? Now, we heard this morning with Koga that we have the visual cortex, from which you have the dorsal stream for action, and then the ventral stream for object recognition, perception. And the hand actions are mostly, the classical view says that mostly it's controlled via the dorsal stream here. And within the dorsal stream, you have two sub-channels. You have a dorsolateral stream here, very specific for grasping movements, and you have a dorsal-media stream that processes the reaching component, so the distance and the direction. When I'm saying that hand movements are really under control of the dorsal stream, that can be, there is a lot of evidence for that. Now, I think the best and the easiest way to explain you this is to think about illusions. And you may know that illusion, right? This is the Ebbinghaus illusion. So if I'm asking you which one of the orange circles is larger, then you're going to tell me that it is what, the left or the right one? Left. The left, right? So that's the illusion because of the context, because it's surrounded by small circles. Now this is your ventral stream, so the occipital temporal stream, because you are recognizing when there is a bias in the ventral stream, and you're recognizing the left as larger. Right, now let's change things here, and if I'm putting marbles on a table, and I'm asking you to grasp the red marble, and if I'm now measuring, like Jean used to do, my finger aperture, is it going to be biased by the illusion as well? So, what do you think?
No.
No, and this is highlighting the fact that there is a distinction between the dorsal stream and the ventral stream, right? So my finger aperture is going to be the same here. Now, if you're not convinced yet, there is another illusion, which is the size-weight illusion, and that illusion says that if I'm asking you now to pick up the small cube here and the large cube, what you don't know is that they actually have the same weight, they have equal weight, you don't know that. I'm asking you to pick up the small and the large. What you're going to tell me if I'm asking you to estimate the weight, which one is the heavier? Well, you use your ventral stream, perception, recognition, and you're going to tell me that the small is actually the heavier. It's because there is a mismatch between what you expect and what you get. Now, again, if I'm asking you, if I'm measuring your fingertip forces, which force do you use to lift these objects? And I'm putting these little force sensors on top of these objects. Well, same answer, my force is not biased by the illusion. So I'm going, actually, after 2 trials only, I'm going to use the same force to lift these 2 objects, even though you keep telling me that the small one is heavier. So if you use your ventral stream, then you would lift it up with more force. So distinction there. Now, this is the theory, right? And I mean, this maps, these circuits in the brain, that looks, I'm sorry about it, it's a bit messy and it looks like the London Tube map. It's quite complicated, but let's put some colors in here. We can recognize the brain areas that Kruger talked about this morning, so MIP and PMD here, that's in the dorsal stream, the areas that control the reaching components, the distance, the direction, and in blue you have the grasping component. And as you can see here, these are TE areas in the temporal cortex, in the ventral stream, and you can see that you do have these green lines here. These are interactions between the ventral stream and the dorsal stream. And we've heard about concepts as well this morning. And of course, it's very important to use what you know about an object to guide actions. Let's say, for example, you need to grasp a ripe tomato, very fragile. So you better know how fragile a ripe tomato is. What does it mean? The color of it is very red. Otherwise, you don't want to use too much force to grasp it. So there are, this is a very basic example, but there are obvious examples in real life that these two streams, they do actually interact. Now, I'm going to talk a lot about hand shaping. And what I mean by sensorimotor transformation is how do you process visual information such as size and shape of an object? How do you translate this into a motor command? How, based on the shape of an object, you're going to grasp, you're going to shape your hand in such a way that it's appropriate for the grasp. Right. First, just a little bit of explanations of the type of signals that you can find in the brain in these 2 channels in the dorsal stream. We talked about MIP, the medial intrapalatal area. That's an area for reaching movements, and if you stick an electrode here in a macaque brain, what you can find, this is one neuron recorded in like maybe 10, 12 orientations here. So the monkey is doing reaching movements in several orientations, and actually that neuron has a preferential discharge here for a particular direction. So people have said, well, that brain area encodes direction, and you can find the same sort of neurons in the dorsal premotor cortex here, belonging to the same circuit. While let's move to the grasping circuit, and you have the anterior intraparietal area, the ventral premotor cortex. And if you record neurons in these two brain areas, what you will find is a preference for a particular type of grasp or a particular type of object. So if you look at AIP, for example, here you have one neuron recorded in 6 different conditions. This is the monkey grasping plate. This is the monkey grasping the cube, so it's a small, for me that's a cylinder, that's a sphere. But what you see here is that the neuron discharges preferentially for the plate and a bit less for all the other grasp, and certainly not for this one. The same sort of neurons in PMV. That's a neuron that's particularly active for the precision grip between the thumb and the index, but not for a whole hand grasp when the monkey has just to grasp a large cylinder. Now these are recordings, but do we really need these two brain areas for grasping movements? So what you can do to have a causal relationship between these two brain areas and behavior is to induce lesions. So in monkey, what you can use is a GABA agonist, so facilitate inhibition, and that's called, you can inject muscimol. And when you inject muscimol, for example, in the ventral premotor cortex, what happens to the monkey is not here. The monkey has to grasp, has to push on a little switch inside of a hole, and the monkey is able to do that, but after muscimol injections, you can see that the monkey is not able to perform individuated finger movements. Precision grip is not able to do it, and the monkey becomes very clumsy. So we can say that we need these brain areas for grasping movements. This is monkey studies. These are monkey studies. Now, can we do the same analogy with humans, right? So recordings, it's a bit difficult even now to do recording in humans, but you can use brain imaging. And here you have, this is a meta-analysis of activations found in AIP when subjects grasp all sorts of objects in a scanner that activates AIP very easily. You also have activations in the ventral premotor cortex. Now, lesions. To find out the causal relationship between these brain areas and behavior. You could look at stroke patients, and based on the lesion, on where the lesion is located, then you could try to see, okay, what are the movement deficits in these stroke patients. Now the problem with stroke is that the lesion is often very large, and here we are talking about brain areas that have 2 centimeters or even less of a diameter. So what we can use is transcranial magnetic stimulation. And I'm going to explain you what this is, but basically we are inactivating very focally one brain area. We are measuring behavior, and based on the deficits that we induce, we can then infer back what's the causal role of the brain area that we are perturbing. Right, so I've used transcranial magnetic stimulation. What this is, what is it? So basically it's a coil that you apply on top of the subject's head, and you discharge a very high current into that coil that generates a magnetic field, which in turn, that magnetic field generates a current into the brain. And that current is going to depolarize neurons. So that's not painful, it's very transient as well, and people have used it a lot since 1985. Right, this is the one study published in Science where they have recorded neurons while they were using TMS. And this is just to show you that TMS can be used as a technique to inactivate brain areas, to mimic brain lesions. So what they've done in the study, so this was an anesthetized cat, and they were stimulating V1. TMS occurs here, so before TMS, what the cat was presented with a stimulation a visual stimulus, and of course, if you measure neurons in M1, they are firing a lot during that stimulation. And before you can see, this is the resting state of that neuron, and you have a very low discharge rate. What's happening after TMS, 4 seconds here of a repetitive train, well, you can see that at rest, that neuron discharges a lot, but discharges also much less during presentation of the stimulation. So what you can say is that you are affecting the normal working of the brain area. So you somehow induce noise, and this is why we can say that TMS can be used as a way to perturb the normal working of the brain area. I can give you more details if you want later on. Now, very briefly, 2 studies that we've done that look at the causal relationship between brain areas and behavior. One for reaching and one for grasping. So that reaching study here, basically you have a cursor near the center, and what you have to do is go as fast as possible to one of these 20 targets. And we purposely had many directions and many amplitudes because the reaching circuit controls amplitude of movement and direction. What we found by stimulating, we stimulated the left and the right MIP. It's very transparent here. This is the control condition without stimulation, and we measured the initial movement direction. Here I'm only showing these 4 targets, 45, well, the 4 quadrants, the 4 corners. And you can see that cannot see much, but the initial movement direction when you have no stimulation is pretty much precise. If you average 20 trials, you have no bias, and the standard deviation is very small. Now, if I stimulate the left parietal cortex, what happens in red here is that if you average 20 trials, let's say, there is no bias in the average direction, so on average the subject is still spot on the target. But what's happening is that from trial to trial there is a lot of variability. So what's happening is that you have perturbed the encoding of the target direction into a proper wrist direction to get to the target. And the same was true for the right PPC TMS for the left targets. So there is a causal relationship between these brain areas and encoding movement direction. Now, the same for grasping. Here, subjects had to grasp and lift that small object here that was mounted with 2 force sensors, one for the thumb, one for the index finger. What you can do with that object is measure where subjects place their fingers on the object. And here on the top, I'm showing in blue the thumb and in red the index finger. So the 2 graspable surfaces are overlapping each other. So you basically turn the object like this. And all the dots are 20 trials, I believe. And the ellipse is the variability, is the confidence interval. Then you can also measure forces. And what we've done, for example, you can TMS one of these brain areas I talked to you about. If you TMS AIP, what you can see is that, well, again, the variability of your index and thumb finger placement is much bigger. That means that you are perturbing your hand shaping. Now, at that point, scientists were able to stick labels on different brain areas. And here you can see you have a lot of labels. So that brain area does this, that brain area does that for reaching movements, for grasping movements, for all sorts of actions really. So we ended up being, with actors. We know who they are. But we don't know how they talk to each other. And it's very important to understand how brain areas talk to each other, because this is how visual information is transferred into the motor cortex, the primary motor cortex M1, to generate a motor command. And this is what I'm going to show you now. We really looked at how AIP is connected to the ventral premotor cortex, and in turn is connected to the primary motor cortex, which ultimately is connected to hand muscles. For that, again, we used TMS. But we used another technique. We used TMS as a way to assess corticospinal excitability. So how excitable your cortex is. And to do this, we apply a single pulse. It's just 1 millisecond. You depolarize neurons, you have a descending volley, and you can measure a hand muscle. You can measure a hand muscle twitch. And if you put EMG electrodes on the muscle, well, you can have these sorts of bipolar MEP, motor evoked potential. And if you measure the amplitude of that, that can give you an idea of how excitable your cortex is. Now, it becomes a bit more complicated. You can use two TMS coils, and it actually looks. You use two TMS coils, and you look at what's the effect of stimulating the blue area a couple of milliseconds before stimulating the red area. And if you have an effect on your MEP, here, let's say the MEP is now smaller, what you can say is that the interactions between these two brain areas are inhibitory, right? You are following me. And this is how it looks like on the subject. Now, what I'm going to show you now is the ratio between the double pulse and the single pulse. And if the ratio is below 1, that actually means that there is inhibition between two brain areas. What we've done is looking at what's happening when the subject is grasping with two fingers a small plasticine cube using a precision grip, or when the subject is grasping a tennis ball using a whole hand grasp. And we made sure that the force exerted by the muscle that we were measuring, the MEP, was the same. So the only change was basically the hand shaping. What we found is that, so again, the descending drive, the force output is the same. What we found is that the interactions between these two brain areas are different when the hand shaping is different. You can see here, red is for precision grip, and blue is for the tennis ball, the whole hand grasp. So, without going too much into the details, what it means is that these two brain areas talk to each other in a different way when the hand shaping is different. That actually means that what they say to each other is related to the task. I want to say generally in here because now we are going to look at a reach to grasp task where the subjects were presented with either a pen for a precision grip again, or a large disc that you have to grasp with the whole hand. And what was interesting with this sort of task is that the motor command is very specific. And the idea was that if the motor command is very specific, then let's try to see how the brain is generating these two very different motor commands. So again, for the pen, you need a lot of first dorsal interosseous muscle, so you need a lot of that pinching muscle here. But you don't need the ADM, abductor digiti minimi. You don't need that muscle to open your hand. You need that muscle when you do a whole hand grasp. And this is what's highlighted here for one particular trial. This is EMG. When you grasp the pen, you have a lot of FDI activity, pinching activity. The ADM remains silent, and the opposite is true for the whole hand grasp. You have a little bit of FDI because you need to pinch a bit, but you also have a lot of ADM because you need to open your hand to grasp the disc. Now, what we found out is that the interactions between so I'm concluding after this. The interactions between these two brain areas are grasp specific. If you look at the FDI, it was active when you had to grasp the pen, but not when you have to grasp the disc. And conversely, looking at the potentials in the ADM, they were more active when you had to grasp the disc rather than when you had to grasp the pen. And again, I'm emphasizing that this is all happening during visual presentation. You are not yet moving. That actually means that these two brain areas talk to each other in a grasp-specific way. And that was, I don't want to go over too much. Thank you.
Extremely interesting keynote talk.
Do you have any model of how the skin sensor enters the brain, just like the model of the visual cortex? Can you talk about tactile?
Yeah, that's of course important, because when, let's say, you grasp a glass of water, it's a half full, so you can guess how heavy it is. But then once you touch it, your mechanoreceptors are going to tell you, how heavy it is, and then you can refine your your estimation of weight based on your feedback. What we've done is we used TMS to look at whether, just before you touch, whether you open the door, whether you increase the gain to that mechanoreceptor information, and actually you do. So that actually means that you can, all the way up to the primary motor cortex, you do have processing of feedback. That was important for us.
We have to move on, but Mark is staying for the dinner, perhaps? Yes, so there will be chances for folks later. Thank you very much. And now we have Giorgio Meza, that